Hi everyone, so my topic is CRISPR therapy and how it helped save a baby named KJ. So for this topic, most of my information came from an article called First Patient Treated with Personalized CRISPR Therapy Developed in Just 6 Months, and the article was written by Andy Murdoch. So, to give some background information, CRISPR is a technology that allows scientists to edit DNA with precision. Baby KJ had a genetic disorder caused by just one single letter change in his DNA, so literally just one wrong letter. Scientists used base editing, which is a very precise form of CRISPR that fixes just one DNA letter instead of cutting the whole strand. KJ's faulty gene affected the liver, so this treatment used lipid nanoparticles, which deliver the editing tools directly into liver cells. There were 5 researchers who worked closely on this case with KJ. They were IGI researchers from Berkeley's Innovative Genomics Institute. You can see on the picture on this slide a picture of two of these researchers with baby KJ. So babies with this gene mutation that KJ has have a 50% mortality rate. So they had to develop this therapy pretty quickly. This team had to move incredibly fast because the baby's health was at risk. The first thing they had to do was find the best version of the base editor for KJ's specific mutation. And then after that, they had to package the editor into RNA molecules inside lipid nanoparticles. And then everything had to be tested, and the scientists were constantly having to ask themselves questions such as, does this therapy reach the right cells? Does it fix the mutation? Does it stay safe for baby KJ? They had to produce a high quality clinical grade version, which is usually one of the biggest hurdles when it comes to gene therapy. So the picture on this slide is a visual of DNA strand being edited. It kind of gives an idea of what that kind of looks like when a DNA strand is edited. And then the next part of this process was safety testing, of course. One of the coolest things is that they were able to get access to KJ and his dad's full genome the very next day, which made the safety testing far more accurate. So the IGI team then ran a detailed test called Change SEQB, which checks the entire genome for accidental edits. This test takes about 2 weeks in the lab, and it cannot have any mistakes. If there's a mistake within that 2 weeks, essentially they have to restart, fix that mistake, and that puts them another 2 weeks or more behind. So the researchers were feeling a ton of pressure. They even worked through the 2024 holiday season because delays could have put KJ at risk, and they were just working against time to make sure that they could get all this safety testing done and approved for KJ. This picture on this slide shows KJ with those 2 researchers again, and then with KJ's whole family. And then after all of the safety testing, the next step was getting FDA approval. So after all the testing, the team submitted the safety data to the FDA. This typically takes months to get approved, but luckily in this case, the therapy was cleared in just 1 week, which is great because it just put them that much farther ahead in the process into helping KJ. And not only that, but it showed that a fully personalized CRISPR treatment can be created in as little as 6 months from start to finish, because after all of the developing and safety testing and FDA approval, it only took them about 6 months to get this treatment done for KJ, which is a lot quicker than they originally had thought it was going to take. And then the picture on this kind of is another visual that shows before and after with CRISPR gene editing therapy. And then moving on to the results for KJ. KJ had great results from this CRISPR therapy. So far, KJ had 3 doses with no serious side effects. He can now tolerate more protein, which is something that he could not do before. And he's needing fewer medications than he was on before this therapy. He even recovered from normal childhood infections without the dangerous amino buildup that used to happen for KJ. So overall, KJ's health has significantly improved from this CRISPR gene editing therapy. And on this slide, you can see a picture of KJ after, and this picture was taken after he had received those 3 doses. So why does CRISPR therapy and this case about KJ matter? Well, first of all, this case was a huge milestone for CRISPR medicine. It showed that personalized gene editing isn't just possible, but that it can be done quickly, safely, and responsibly. And the future is looking very optimistic. Scientists are hopeful that this approach will eventually become a model to help many more families with rare genetic conditions, not just the condition that KJ had, but this could potentially help many other individuals who have other conditions caused by gene mutations. And overall, it shifts CRISPR from something used on groups, which is what was previously thought of it, to something that can be tailored to one single patient. This picture, I liked it because it kind of can be interpreted a few different ways, but I see it as like the pathway to CRISPR treatment helping change the world. It shows a scientist walking down a path, and that's exactly what I think CRISPR would do. I think it's going to have a significant effect on patients from here on out, especially with how successful KJ's story was. And these are all my citations. It includes the article I got most of my information from about CRISPR treatment and baby KJ, as well as the citations from where each of the pictures on my slides came from. I hope you enjoyed this presentation, and I hope you have learned something meaningful about CRISPR treatment. I truly think that this case for KJ was a huge milestone for CRISPR medicine and an enormous breakthrough. And I'm super excited to see how scientists are going to keep developing this and using it on future patients, and seeing how it's going to help change many lives from here on out. Thank you for listening.